Assalamualaikum and peace be upon you. My name is Afiq Dania bin Zaki, student ID number 2022-497-654. For our video presentation in CHM 256, we will be showing you about chromatography. Our title is the identification of caffeine and paracetamol in a pharmaceutical tablet. We will be referencing Panadols, which are usually used as painkillers. Let's begin with the introduction. What is chromatography? Chromatography is a technique used to separate different components in a mixture. It relies on the principle that different substances will move at different rates when dissolved in a solvent and pass through a medium or a surface. For our assignment, we will be making use of two chromatography methods, which will be discussed later. Next, our theory on why it works. As chemicals in a mixture are only combined or bonded physically, each component will have differing separation rates when passing through a surface, as they are affected by the partition coefficient. The various components of a mixture tend to have varying aspects affecting their stationary phase, causing them to be retained for differing periods of time, based on how they interact with the surface they're on, or in, such as capillary tubes, which changes their apparent velocity in the mobile phase, separating them from the rest of the components. Thus, the individual components of the mixture are carried out when the solution is in its mobile phase, while moving through a system with a material called stationary state fixed or affixed. As separation rates relies on differential partitioning between the mobile and stationary phases, the subtle differences between the compound's partition coefficient causes a differential retention to the stationary phase by the mobile phase itself, and further affecting their separation. Hence, it can be used for the separation, identification, and determination of the chemical components in a complex mixture very accurately. In our research, we make use of caffeine and paracetamol as our mixture.
My team conduct our research of the identification of caffeine and paracetamol in a pharmaceutical tablet by research in a journal through online method in order to find the chromatography method for caffeine and paracetamol in a pharmaceutical tablet. Next source is from the website through online method too. Lastly is from the book that we get from the library.
Identification of caffeine and paracetamol in a pharmaceutical tablet, for example, a resolve. First thing first, we are going to dissolve a small amount of pharmaceutical tablet in appropriate solvent to prepare standard solution of pure caffeine and paracetamol. For stationary phase, composition of silica is formed of silicon dioxide with a porous structure that provide a large surface area for adsorption. For mobile phase, a solvent system of ethyl acetate, methanol, and water in the ratio 1.5 and 0.5. Ethyl acetate is moderately polar solvent. The function of ethyl acetate is for dissolve a wide range of organic compounds and help in eluting polar and non-polar compounds. Methanol is a polar solvent. The function is for methanol increase the polarity of the solvent system, which is its aids in dissolving polar compounds and enhance the interaction with the silica gel. Water is high polar solvent. Water further increase the polarity of the solvent system and help in separation of very polar compounds. For the process, spot the sample and stand it on the baseline of silica gel coated TLC plate. Develop the plate in the solvent system. Visualize and analyze the spot under UV light or iodine vapor. Calculate RF values and compare them with standard to identify the compound. Formula for RF is equals to distance traveled by solvent front over distance traveled by the compound. For example, if the solvent front travel x cm and the spot for caffeine and paracetamol travel 5 and 6 cm, RF for caffeine is equals to 0.625 and RF for paracetamol is equals to 0.75. By comparing these RF values with the standard, you can confirm the presence of caffeine and paracetamol in the pharmaceutical tablet.
Assalamualaikum, my name is Adam Danish Bakir. Next is the identification of caffeine and paracetamol in a pharmaceutical tablet in manufacture. The process to manufacture caffeine and paracetamol in pharmaceutical tablet involves two steps, which the second step consists of two methods. First step step is to prepare the sample. This is done by crushing the tablet into a fine powder in order to make sure that it is homogeneity. The powder is then weighed to a suitable value and have it soft in a suitable solvent. The suitable solvent can be methanol or water or any other kind of solvent. Next, the second step consists of two steps, which is the first method is called high performance liquid chromatography, or it is called HPLC. This method requires an equipment HPLC system with a UV detector. Then standard solution of caffeine and paracetamol with a known concentration is prepared. The sample is then injected into the HPLC system and is set to a suitable flow rate and temperature. The HPLC is then run and the chromatogram is recorded. The result is then judged by comparing the retention times and peak areas of the sample with those of the standard to identify and quantify caffeine and paracetamol. Second method is called ultraviolet TLC chromatography. This method requires TLC plates and a UV lamp for detection of caffeine and paracetamol. A standard solution of the caffeine and paracetamol is prepared and the TLC plate is prepared too. Then a small amount of sample of is then placed on the baseline of the TLC plates by using the capillary tube. The TLC plate is then placed in a development chamber containing the mobile phase and the solvent is then allowed to rise up on the plate. After the solvent is done rising, the plate is then allowed to dry. The plate is then allowed to dry in a fume hood. The spots of the sample is then determined under the UV lamp. The distance travelled by the spot and the distance travelled by the solvent is measured. The identity of the caffeine and paracetamol is found by calculate the value of RF. That's all from me.
For identification of caffeine and paracetamol in a pharmaceutical tablet in industry, we use ultraviolet visible spectroscopy (UV-Vis). For sample preparation for UV-Vis spectrography involves solution formed with a higher absorbance wavelength than the analyte, dilution to align with Beer-Lambert law, and accurate preparation using volumetric glassware and digital pipettes. UV spectroscopy has many applications, including quantifying organic compounds, transition metal ions, and biological macromolecules, monitoring structural changes in DNA and proteins, quality control in the pharmaceutical, food and chemical industry, detecting and quantifying analytes in vital analysis, serving as a detector for various chromatographic techniques. For infrared spectroscopy, sample preparation, solid samples, crush the sample with a molding agent and apply a thin layer on a salt plate. Liquid samples, keep the sample between two salt plates, and gaseous samples, use a sample cell with a long pass length. For applications, organic and inorganic chemistry, IR spectroscopy is used to determine functional groups as molecular structures. Quality control, IR spectroscopy is used in quality control in various industries such as pharmaceutical and food analysis. Biological research, IR spectroscopy is used in protein characterization and nanoscale semiconductor analysis. For conclusion of principle of chromatography, chromatography is a method of separating molecules in a mixture by interacting with a stationary phase and a mobile phase. The stationary phase is typically a solid or liquid, while the mobile phase is a liquid or gaseous. The components of the mixture interact differently with these phases, leading to their separation based on molecular characteristics. The retention time of each component is a key parameter in chromatography, used in various fields like industrial processes, pharmaceuticals, and biochemical laboratories.